Okay, now we can record. Yeah, awesome. Okay. Dooply. And there we go. Oh, I'm right in the middle. Okay, so we left off with this equation here. And actually, I should probably unshare screen because I'm going to write on the board real fast. But it's really not a big challenge in here, but I just want to go through and kind of derive this very quickly. So I'm going to pause the screen sharing. Yeah, okay. Hopefully you guys can see me on online, right? Yep. Hope. Okay. So we're starting off with this awesome equation up here. All right. And how much can you guys see up there? Yeah, I don't know if you guys can see that very well at all or not. But we're also on this, was it slide 48, I believe? Yeah, slide 48 as well. And we're going to kind of follow along. And we start off with a simple, fairly simple description of our acid dissociating into basically a proton and a conjugate base. Okay, so that's what we've got up there. All right, so we can describe this dissociation constant as our Ka. We've got that up there. Our Ka is basically just the concentration of our products, our H plus and our A minus, over the concentration of our undissociated acidic form. Okay, so if I rearrange this, that's what I get in the middle there. The part that's kind of covered up. So I can just kind of solve for H plus. My H plus concentration is basically going to equal my Ka times my HA or my A minus concentrations. Okay, so we can figure out how much proton we have in there. All right, so now the tricky part is we're going to use my logarithms. All right, so I'm really going to take the negative log of both sides, and I'm going to get to this, but the key is I need to recall that negative log of Ka times HA over A minus. Okay. I can separate that out. I can make that negative log of Ka minus the log of HA over A minus. So our logarithms have that distributive property or something like that. So I get to that. All right. No big deal. We're to this equation now. All right. Well, if I look at this negative log of H plus, I can just call that pH. Negative log of Ka, that's going to be my pKa. And now I have negative log of HA over A minus. All right. Again, another log property. If I change the sign here, I can just flip my divisors over here and change that sign. So instead of having negative log of HA over A minus, I'm going to make that plus log of A minus over HA. Okay. And this is how we get to our Henderson-Hasselbalch equation, which we'll be using here in the next couple slides. Okay. All right. And again, just keep in mind pH, just telling us how acidic or how basic our compound is. Our pKa is specific to our particular weak acid that we're utilizing, and we're just looking at the undissociated versus the dissociated portions of the acid, of the weak acid that we're using. Okay. So it's not a terribly bad derivation, but I like to write out these extra two steps just so that everybody kind of sees where it's coming from, right? And to remind you of the funkiness of logarithms working in there. Okay. All right. So we're going to apply this a lot when we make buffer solutions or when we analyze stuff. So let's go back to sharing. Resuming share. Yeah. All right. And we'll move on to the next little slide here, right? Yep. Nope, too far. Go back. Go back one. There we go. All right. So in this case, we have this lovely little example of acetic acid here. Okay. We have a buffer, and we're going to make it out of 0.1 molar acetic acid. And 0.05 molar sodium acetate. Okay, so my sodium acetate is essentially the salt of my conjugate base. All right, So you can see we've got our CH3COONA. Because that's an ionic compound, we know when we drop that into water, this is going to completely dissociate, right? So we don't have to worry about it not dissociating. So we're essentially not going to worry about the Na, and my 0.05 molar sodium acetate means I'm going to have 0.05 molar acetate anion in solution. I'll actually have a little bit more because maybe this will be in there, maybe it'll dissociate, but we're not going to sweat that detail too much. So we're avoiding the complex math. So if I look at this, I can say my acetate anion concentration is 0.05 molar. My acetic acid concentration is 0.1 molar. Right? I know, and I would give you this information. You'd be able to have the access to this information. My pKa for acetic acid is 4.76. So I want to know the pH of this solution. I just plug it into my Henderson-Hasselbalch equation here. Figure out what its log value is. Because my calculation over here is going to be less than 1, we know this will be negative. So my pH should be a little bit lower than the pKa, 4.45. Okay. This is actually a good check to make sure that you have things set up. In this case, we can see that my concentration of the acid form is greater than my concentration of the base form. So I should kind of be tipped more acidic. So my pH should be lower than my pKa. If I have more of the base form, then we'd expect the pH to be a little bit higher because it'd be a more basic solution. Okay. So that's a good way to make sure that your answers make sense. And that's a good question for me to ask as well too. How do you know that you've done your calculation correctly?
So you say the pH has to be lower than the pK?
Yes,
okay.
Because we have a higher concentration of acid than we do of base.
So it's just reverse.
Mm-hmm. Exactly. Then we'd have a higher pH. All right. And that's not too bad. And this is, in a way, this is a little bit of a review of Henderson-Hasselbalch equation. You probably saw this somewhere in Gen Chem 1. But we're going to apply this to our proteins and amino acids because amino acids are acids and bases. Yes?
How did you know that 0.05?
So the 0.05 is my acetate. So that's going to be basically my A minus. Yeah. And then the acetic acid is the HA form. Yeah. Because it's going to be protonated. Okay. So again, that'll go from that. The acid form will always be the HA, and the conjugate base will always be the A minus. Good times, right? Okay. All right. So how do we use this? Okay. We do buffer solutions. So we have a lot of biological buffer systems out there. We think about our bloodstream. We want to maintain this intracellular pH for all the different organs, and they all have an optimal pH. So your blood pH is different from your gut pH, which is different from your lung pH, and your bile ducts and neural and all that stuff in there. So there's an optimal pH, and the proteins and other stuff that interacts best at these pHs. And if you stray too far from that pH, you disrupt the electrostatic interactions, and you die. My morbid take on life. All right. Our solubility will depend on the proton donors and acceptors. When we would do acid-base, the titration of amino acids coming up in the lab, a couple of amino acids are not terribly soluble, so I would just put in a little more acid in there to enhance their solubility. We're going to make sure everything is protonated so that we can make it more soluble. All right. We also have to look at carbon dioxide as a gas that can be solubilized in the bloodstream. Combine that with water and you get HCO3, really H2CO3, but then you can donate that proton. So you have this carbonic acid solubility, and we'll see this when we talk about the Bohr effect coming up later. Okay. Buffer solutions are mainly based on phosphate, bicarbonate, and a little bit on histidine. Okay, so our phosphate concentrations are typically going to be in millimolar range. I'm going to know that our bicarbonate is how we buffer our bloodstream, and histidine to keep things We can also make other ones in here. It's really funny because Lehninger likes to talk about HEPAs and pipes and chess, and those compounds, I think I've used HEPAs twice. I just didn't make buffers that utilize these. I used phosphate buffers primarily. But anyway, we have a bunch of thousands, literally thousands of weak acids and bases we can choose to make buffers out of. All right. So switching gears just slightly, again, we're looking at water here, and we know that water is both an acid, a weak acid and a weak base. So water can actually bind to proteins. You can kind of see our little blue globs of water around here surrounding it, and then the participation of water, the participation of the solvent, can become critical for the function of various proteins. What are we looking at here? I think this is hemoglobin. All right. So in order for our hemoglobin to work, we have some water molecules that have to assume specific positions within there. So if we look in here, we have this kind of little cleft or channel where we'll see about five or so water molecules that are going to line up in there and form hydrogen bonds with these various residues. And then these water molecules will be stably positioned in there, and they can help with the transfer of electrons and protons. Right? So what do we have here? Yeah, just don't want to say anything else about this. Yeah, okay. Yeah. All right. So somewhere down here, where's our heme group? Heme kind of stuck up here. So we've got a heme inside our hemoglobin, and then one of the little tails comes over here and interacts with this glutamine, which is helped stabilized by these water molecules. And this becomes important for electron transfer in our hemoglobin and all that good stuff in there. Okay. What else do we have in here? Here we have a sugar binding, a ribose binding protein. And again, we can see that there are a couple of water molecules that have to sit in these particular positions to optimize the binding of a ribose. Okay. So our water, even though it's primarily, we're going to think of it as a solvent, it actually has a functional role in a lot of the activity that we're going to talk about. All right, so there we go. It's chapter 2, not too bad. Intermediate forces, properties and structures of water, weak acids and bases. Not bad, all right? So our first 2 chapters are essentially, screen sharing paused, okay, yeah. Let me get rid of this one. No, let's pause the screen. Okay, hold on. Let's find a new one. Yes.
How would our test look like?
So in theory, I'm going to have a series of short answer questions for you guys. So it'll probably be a couple multiple choice thrown in there. Maybe, maybe not. Yes. So normally for this first exam material, you'll have questions like, okay, what are the structures of the particular proteins? Oh crap, got the wrong one in there. Again, hold on. Or I would say figure out what the pKa of this acid is, or what's the pH of this solution, or what concentration of this acid would you need to make a buffer at this pH, something like that for these types of problems. But I try and theory craft a little bit as well too. Okay, is this the right one? Yes, this should be the right one. No. All right. Tell you what we're going to do. We're going to find my PowerPoints. We're just going to close this one. There we go. Now we're going to share screen. Okay. Awesome. There we go. Okay. And as we get closer to the exam, when we start finishing up this chapter, I'll have a review session as well too, where I'll talk more about the details of what will be on the exam and how I'll present it. It'll be interesting this semester. Got to revamp my exams more than I normally do. I don't know if that means you're going to have more questions or fewer questions. I don't know if either one of those is good. Okay. So if we look at chapter 3, we're finally getting into real biochemistry now. This is where the biochemistry really begins. You know, we talk about all this foundational stuff and go through our backgrounds and all, but now straight up biochem. So here we're going to talk about amino acids and small peptides. We're going to look at the ionization of our amino acids because we're going to have lots of ionizable protons on there. So their acid-base behavior, we'll talk about that. And then we'll take a little break from that and talk about different ways we can characterize them as well too. Okay. And that's kind of neat and sometimes ties into lab. All right, so what are our proteins? They're our main agents of biological function. If something's happening in your body, it's almost always going to be associated with proteins that are doing it. So we can have proteins that act as catalysts. We can have things like enolase or DNA polymerase, which are facilitating reactions. We can have transport mechanisms. Think of hemoglobin transferring oxygen from the lungs to the tissues, or lactose permease transporting lactose across the membrane. We have a lot of structural proteins, collagen and keratin. You think about your hair, keratin fibers make up a huge amount of that, collagen for your skin and extracellular matrix, fibrinogen as well too. Motion, if we're in any way moving, we have muscle activity in here. We can have our actin-myosin connections going on. So, you know, if something's happening, it's probably got a protein, all right? I always like to keep this picture in here because here we have a rhinoceros with its horns. Anybody know what the horns are made of? Keratin. So just like in your hair, your skin, or your nails. So there are something like 86 different keratins, maybe more than that. There are particularly human hair keratins, which I'm interested in because we're isolating human hair keratins to help stimulate the proliferation and movement of Schwann cells to help initiate regeneration of peripheral nerves that have been damaged or severed. Could you do that with a rhinoceros horn? Maybe. Now this is where you start getting into cultural things. If you go around a lot of different cultures, you know, rhinoceroses are endangered because people want to cut off their horns, right? Same, you got elephant tusks carved for the ivory, but a lot of cultures will have traditional medicine where you would use, say, rhinoceros horn or elk antlers or you know, but various animals in there. There may actually be something to all this. So yeah. I'm not saying go to the zoo and, you know, steal the rhinoceros's horns, but you got nerve damage anyway. So if we talk about proteins, let's start from the very, very basics. All of our proteins are made up of amino acids, okay? So our proteins are basically linear heteropolymers, okay? So we have 20 basic building blocks, or 20 amino acids that you should know the structures of by the end of this course. I had them all memorized, and now I don't have to memorize them anymore, so I have about half of them memorized, you know, and others I can look up. I'm close on the rest of them. Now it's all application, yeah. But you gotta make flashcards. I think there might be a link in the supplemental material to maybe kind of like it's either a Quizlet or something like that where you actually already have pre-made flashcards. But I know, yeah, I've had students and other people I've kind of mentored study for, say, the MCAT or med school, and basically get like index cards, write the structure on one side and the name, the abbreviations on the other, and all that good stuff in there. So it's good to have those memorized, right? Our amino acids have a bunch of properties that make them useful for carrying out biological activities. So for one thing, they are linear, right? So we're not going to see any branch chain proteins coming out there. We can add stuff and we can decorate, you know, modify proteins, but they're going to be linear. Our heteropolymers, we got 20 of them in here. So that ability to polymerize is important. So we have growing proteins. I think I mentioned like membrane ruffling and the movement of the actin cytoskeleton, where you actually have it grow forward to pull the cell along, and it disintegrates at the end. We have a lot of acid-base properties with our amino acids. There's a bunch of different physical properties and a bunch of different chemical properties that we'll talk about here. And we can classify our 20 amino acids based on some of these different properties, which helps make them a little easier to memorize. Okay. So amino acids all have some common features in there. We're going to have the central alpha carbon, right? Connected to that, we're going to have an amino group, and we'll have this carboxylic acid group. So here's our amino acid, that's how we get our name. and then there'll be a hydrogen coming off of the alpha carbon, right? And then we have an R group down here. And this R group will be different for each different amino acid. The simplest amino acid is probably glycine, where our R group is just another hydrogen, and everything else is something different. You can see, as long as this isn't, hydrogen, we have 4 different substituents attached to our alpha carbon. So we have a chiral carbon there. Our alpha carbon will be chiral, except for glycine. All right. So the alpha carbon always has 4 substituents. So it's always going to be in this tetrahedral arrangement. All amino acids, except for proline, and we'll see why, because it's got a funky structure here. It's going to have this acidic carboxylic acid group, basic amino group, and that alpha hydrogen in there. And that 4th one is something else. Okay. Now for proline, that R group actually comes back and connects to one of the others in there as well. All right. So we have like a circular structure, which actually makes proline really useful in some cases, which we'll get to in like chapter 4, I believe. So all amino acids are chiral except for glycine. Proteins only contain L amino acids, right? You can find L and D amino acids in nature. D amino acids do occur, but we only incorporate L amino acids into proteins. Okay. A great exam question might be, hey, what happens if you eat a bunch of D amino acids? So, and the answer to that is actually, I know, explicitly in the Lippincott's Illustrated Review, and you can find that pretty easily elsewhere. All right. But if we look at this, if we go back to how we draw structures, we can do our dash and wedge model in here, have L-alanine and D-alanine. I won't make you draw the dash and wedge stuff like that. That's a little too much, but we can, kind of, it's not really a Hayworth projection because it's not a sugar, but we can have this consistent drawing here where this will be our L-alanine. If we kind of flip this over to the mirror image, we get our D-alanine. But I'm not going to sweat that too much because we're only pretty much going to talk about the L amino acids. I always take a little moment to go inside here. There's one professor in the grad school I went in. He studied L-arginine. L-arginine was the answer to every one of his questions ever about everything. And what was really funny is whenever a student would present, on a topic for a seminar series, he'd always ask him a question, and it always dealt with L-arginine, and he was very, very long-winded. And most of my fellow students were like nervous because he would talk so much, and he'd just turn everything to his research. So I was like, you know what? I'm going to handle him because I knew going in that he was going to ask this question, and it was going to get long-winded, and he's going to turn around this way. And so I figured out how to say, well, that's actually really interesting, but that strays from the topic that I'm talking about here, but I'd be glad to talk with you more about that after the seminar. So when you guys get the senior seminar or presenting stuff or have to give talks like that, how to handle difficult audience members, useful. All right. Anyway, back to our amino acids. We got a bunch of chirality here. We can classify our amino acids based on their polarity and their charge. Okay. So common amino acids, we typically break this down. We have 20 of them. If I just said memorize 20 structures, it'd be like, oh, that's a pain. So we break it down into 5 categories. So you've got 2 negatively charged, 3 positively charged, 5 that are uncharged but polar, 3 aromatic, and 7 nonpolar. Okay? So that's not bad. You can start with these 2 and say, oh, I'm going to memorize the 2 negatively charged ones. And then I'll memorize the 3 positively charged ones. But a lot of it is just drawing the structures over and over again or working those flashcards. I don't think neural networks for humans have changed that much yet, but there are studies from the past that suggest people, when you write something down, it makes a connection with your brain and you retain it longer. So that's why I always kind of recommend writing, drawing out these structures. Digitally, I don't know if you can probably get the same thing with, like, say, a stylus and digital, but I don't think there are updated studies now for everybody who purely types. But anyway, I recommend all that good stuff in there. All right, so guess what we're going to do? We're going to look at structures. Yeah. So let's look at our 7 aliphatic nonpolar ones. All right. We start off with our simplest one, glycine. Probably the easiest one to memorize because it's just another hydrogen in there, right? So here's our basic structure. Everything's going to look like this. All right. Alanine, we just put a methyl group instead of our hydrogen in here. I'm going to skip proline for a minute. Valine, that makes a little V shape with methyl groups, right? Leucine, we just stick an extra one in there. Isoleucine, well, instead of having the V at the end, we just have this little T shape in here. So it's the same number of constituents in the side chain as leucine. It's just rearranged slightly. And then methionine gets a little bit trickier because we have this sulfur group in between our three methyl groups that we have here. Right? And then proline is weird, okay? Because again, we just have three methyl groups, but we actually form a ring structure back with the amino group. So proline's an odd one, but it has really specific functions. And you're going to see, proline's at relatively low concentrations most places, except in a few particular proteins where you see this huge spike in the concentration because it helps make bends. All right. So those are the nonpolar ones. Okay. There's 7, and they're generally not too bad. With a little practice, you can kind of get these. So that might be like one day and memorize the nonpolar ones. Get tattoos of them on your exam. I wouldn't recommend that. All right, if we go through our aromatic ones, I always found these a little more challenging just because they're bigger. We have our aromatic ring structures in here, but phenylalanine, probably not too bad. Methyl group and a ring structure. Tyrosine looks almost the same as phenylalanine, except we put a hydroxyl group at the opposite position in there. Okay, and then tryptophan. I'm like, oh dear God, what's the structure of tryptophan again? Because it gets a little crazy. We got our methyl group, and we have this double ring structure here with a nitrogen in it, and it's like, oh Lord. So that was one of the harder ones for me to memorize. So of course, guess what? One shows up on an exam, right? Yeah. So tryptophan has that crazy structure in there. Okay. We'll see functionally, tyrosine is going to have a function similar to serine and threonine because these hydroxyl groups have ionizable side chains here, and they're great targets for phosphorylation. So we can kind of use that phosphorylation as a signal to turn on or turn off a process. So good regulatory targets. Tryptophan, what do you guys know about tryptophan? About Thanksgiving dinner, eat your turkey, it's full of tryptophan, you get sleepy and you fall over. Tryptophan is supposed to be somewhat somnolent. It's soporific, I guess would be the adjective form. Not really true. Yes, I guess melanin has a stronger effect in there. So most people get tired, not so much because of the tryptophan, but because of just eating a large carbohydrate-laden meal that makes people sleepy. If you go on into Biochem 2, we'll talk about metabolism and postprandial. But because turkey has higher amounts of tryptophan, people tend to think that makes you sleepier. Yes?
So how did you say that people remember how to memorize the structures?
Repetition, just kind of like drawing them over and over again a few times. Like a lot of people who've successfully done this, I know, make flashcards. So if you make your own flashcards in practice, just kind of like draw your own practice in there. When I was studying for my cumulative exams in grad school, I would just see, I'm going to start off, I'd start off my day of getting into studying by just seeing how many I could draw. And if you do that for a couple of days, you got them down pat. Yes?
So we have to remember the names and the structures.
Yes.
For this class.
Yes. And the abbreviations that'll be coming up. Yeah.
What was the question?
I will get to that, yes. The previous question was, do we need to memorize the structures and the names? And the answer is yes. And the abbreviations. So we will see some functional stuff that'll come up here. Most of that will be headed on into the next, in like chapter 4. So we'll get a little bit, we'll talk in general here. Biochem 2, we'll talk about this specific residue having a particular function at certain areas there. But just having a good idea of their general acid-base properties. And we'll see that coming up here in a bit when we talk about isoelectric points and stuff. All right. If we look at our polar ones that are uncharged, this is our second big group. So we've got 7 out of the way now. Here's 5 more. So after this slide, we'll be over halfway done. We've got serine and threonine. Again, these are kind of related because here you have a methyl group with a hydroxyl group on the end of it. And threonine, you've got kind of the same thing here, except you have another methyl group attached there as well. Okay. So the big key here is these hydroxyl groups are polar, and at physiological pH, usually around 7, they will be protonated. So that's why there's no charge. Okay. The reason we're drawing our base structure as NH3 plus and COO minus is that typically at physiological pH, this amino group will be protonated and the carboxylic acid group will be deprotonated. When we talk about the pKa's of our amino acids in a little bit, we'll see why that is. You know, this proton comes off really easy. This amino group holds onto its proton overall. And you can see we have a positive charge and a negative charge, so our overall charge is 0. Okay. Cysteine has a really cool function. So here, cysteine is really similar to serine, except instead of a hydroxyl group, we have a sulfhydryl group. Okay. And again, our sulfhydryl group will play particular roles in cross-linking. So we can form a disulfide bond between two cysteine residues, and those can hold, you know, we actually have a physical covalent bond there, so that will actually hold proteins in a particular shape. I'm just going to go back up one slide for just a second to compare this with methionine. All right, up 2 slides, I guess. So you see methionine here, we have the sulfur in the middle, all right, and it doesn't pair. It doesn't, you know, make a sulfhydryl bond or anything like that. And that's because it's kind of trapped in between those carbon groups. With this one being on the end, our hydrogen becomes ionizable, so we can kick off that proton and see that sulfhydryl group. So we have a more active functional group with our sulfhydryl group than just substituting in that sulfur in the hydrocarbon chain. The other 2 that we have down here are asparagine and glutamine. Okay, so asparagine and glutamine are a little funkier. I actually find aspartate and glutamate easier to memorize first and then switch to memorizing these. But here we have this carbonyl and this amino group attached. And for asparagine, we have 1 methyl group in there. Glutamine, we have 2 methyl groups in here. So basically glutamine is just a little bit longer than asparagine. All right, so that's those 5. We've got a couple that are positively charged. All right, so lysine, arginine, and histidine. Why are they positively charged? Because, we don't have this one drawn properly, but because they have amino groups in their side chains. So lysine, it's really kind of funky. We got like 1, 2, 3, 4 methyl groups and then an amino group at the end. And at physiological pH, this will be protonated, so we'll have a positively charged amino acid. So arginine gets really funky because you'd say, well, 1, 2, 3, and then, oh, no, wait, we got 3 nitrogens in here. So arginine can be kind of, you know, challenging to remember its structure, but one of these will be protonated. Yeah.
So like the drawing structure, do you get like partial credit, like if you miss one methyl group?
I do. So, you know, I do like to have exams that you guys can write answers to that I can award partial credit for. So, you know, it's a little harder to talk about it, but if this were like a 10-point question, you know, I might have like, you know, 2 points here, 2 points here, 1 point here, and like 5 points for the rest of this, you know. So there is partial credit in there. And the other thing I try and do is if in the past when I've asked for structures, I'd usually pick like 3 or 4, and there'd be one that's fairly easy to memorize, one that's not too bad to memorize, and then one that's like arginine or tryptophan. So it's crazy. Yeah.
So you say it's positive. It's charged positive because of the nitrogen?
Yes, because of the amino groups in the side chain, they're going to be protonated at physiological pH. So you can see we've got 2 positive charges, 1 negative charge. So my net charge here is going to be +1. Okay. And we're going to see how at different pHs we can alter this. Histidine is another funky one. You can say, well, shouldn't that be in the aromatic? Well, it's not quite aromatic because it's just a 5-membered ring. We've got a couple double bonds in there. And of course, this double bond can kind of flip over here. So yeah, we can kind of rotate it around. There is some variability in here. If I just ask you to say, for example, draw lysine, You could draw it like this, but it would be correct to say NH2 or COOH, or have this as being NH2 or something like that. So to get around that, sometimes if I want to be really specific or mean, I'll say draw the structure of lysine at pH 4, and then I have to give you some pKa information about that. But we'll get to that. We'll get to that. I won't be that mean to start off.
So if the pH changed, the structure will change?
Yes, yeah. Or the ionization state will change. Okay. All right, the last two are negatively charged ones, and compared, these are related very closely to asparagine and glutamine. Here we have aspartate and glutamate. We just have this carboxyl group here. So you can see we have something very similar, either 1 or 2 methyl groups, and then a carboxylic acid group here. And at physiological pH, this will be deprotonated. So I've got 2 negative charges, 1 positive charge, so these will be negatively charged at physiological pH. Not too bad, right? And it's like, well, it's awful fast going over all these structures. So, all right. And just to show you again, my aspartate and glutamate. So if we look at asparagine and glutamine, again, our aspartate and glutamate, we would just get rid of our amino group and replace that with an OH group, well, or an O group, really. So I think there are strategies for helping your brain memorize these. I would start with, start with glycine. Maybe start with the hydrophobic ones or non-polar. I always call them hydrophobic. Maybe work to aspartate and glutamate, and then from that go to asparagine and glutamine, and throw lysine in there because it's the other positively charged one. But just find what works for you. Okay. All right. Mean, because I'm making you memorize them, because you're gonna have to have them memorized for exams that you'll see upcoming, whether it's MCAT, PCAT, or first year med school or something like that. And then eventually you can just start using them. All right. If I give my exams online, memorization is going to be kind of funky. So I'm like, we'll figure something out. Maybe I'll just ask you weird questions about them. Not weird questions. Straightforward, honest questions. All right. All right. So all of those that we looked at, we were assuming they were at physiological pH. Okay. But we can isolate amino acids and we can put them in like, a really acidic bath, or, we can stick them in bleach or something. Right? And we can change the ionization state. So our amino acids have all of them have at least 2 ionizable compounds, the amino group and the carboxylic acid group. And then some of the side chains have an ionizable group as well too. Okay. So all of them have this carboxylic acid. We can have that as COOH, or we can abstract that proton and leave behind our COO minus. So we can either have a neutral or negative charge there. The amino group, we can have the NH4 plus. We can abstract that proton to have NH3 and an H plus come here. So we can go from plus to minus. Okay. Alrighty. So, now we're starting to get crazy. Okay. So again, this is a good figure, 3,1 in here. If you look at, say, acetic acid, we saw acetic acid has a pKa of 4.76, and it just depends on that carboxylic acid kind of coming off of here. If we look at our amino acids, like say glycine, our carboxylic acid, its pKa is less than half that, 2.34, and that just happens to be because our other groups that are attached to the carbon, where the carboxylic acid is attached, affects the ionizability of this proton. So here we really just have a bunch of hydrogens and this kind of carboxylic acid all by itself. All right. Here we have some added stress or strain or effects that make this proton easier to come off. Okay, so we can kick off that proton pretty easy. Probably because we have this protonated, well, definitely because we have this protonated nitrogen over here. We can kick that one off pretty easy. Okay. If we look at methylamine, all right, to abstract the proton from methylamine over here in blue, its pKa is about 10.6. All right. For our glycine, to abstract this proton from glycine, our pKa drops to about 9.6. Again, because that carbonyl group makes it a little easier to boot off this. So this slide is just kind of illustrating that our amino acids, the different constituents of the amino acids, help to affect how easily ionizable the protons on the particular amino acid is. We're basically making it easier to boot off these hydrogens. Okay. All right. And that's why we'll see different pKa's for different groups for our different amino acids. And so we just looked at glycine there. If you compare glycine to, say, valine or lysine, you would see different pKa's for those amino and carbonyl groups. All right. So I'm probably going to start drawing some stuff here in a minute. I got to look at my time. Okay, yeah, we're good here. Let's talk about the ionization of our amino acids. Okay. So at very low pH, if we drop something in concentrated sulfuric acid or something like that, everything is going to be protonated. If there's a spot where we can fit a hydrogen, we're going to have one on there. So my my amino group is going to be protonated, and my carboxylic acid is going to be protonated. If I've got a side chain that can be protonated, it will be as well too. If we instead go to a really high pH, a really basic solution in here, then we're going to pull off all the ionizable protons. So my NH3 plus will become NH2, my COOH will become COO minus, and my ionizable side chain will be deprotonated. All right. In between, at physiological pH, if we looked at our uncharged ones, we could see, our amino acid group has a Zwitter ionic form. All right. So we basically have a tipping point. So we're neutral, and then if we pull off one proton, oh, now we're negative. If we go the other way and add one proton, we're positive. So we can talk about this as being a Zwitter ionic form. I'm getting ahead of myself. Let me rephrase that. If we looked at physiological pH of like our glycine, we saw that we wrote the amino group as NH3 plus, and our carboxylic acid group as COO minus. So that basically has both a positive charge and a negative charge that are stabilized on the molecule, but the overall net charge is 0 because that positive charge and negative charge cancel out. Okay. So if we look at something like glycine here, if I have a very low pH, I'm going to protonate it. It's going to have a charge of +1. Okay. So here's my form at the very top on the left. I have NH3 plus, and my carbonyl group is protonated at this very low pH. Okay. So this is its +1 form. If I start adding base to it and increasing the pH, I get in here and I find out that the pKa of my carbonyl group, and the carbonyl group will always be referred to as pK1. Right? For glycine, my pKa is 2.34. So I can actually make a buffer solution out of glycine. Should have brought my Mountain Dew. So no, you can just stop talking. We're in class early. Right. But we can actually make a buffer solution out of pKa, out of glycine at a pH of around 2.3. Okay. So we see that, as we start adding base, it takes more and more base to get a pH change until we overwhelm that capability. All right. At this point, once we've kind of overwhelmed this, we've now abstracted our proton, and we have NH3 plus and COO minus. When everything is in that form, that's our isoelectric point. Okay. So our total charge is 0. So the isoelectric point is when the charge of the molecule is 0, and we'll revisit this a couple times. Right. If I keep going, I can start to abstract the proton from the amino group, and my amino group, which is always pK2, has a pKa of 9.6. Okay, so I'm going between these two forms, and eventually when I abstract all that off, I'm going to have NH2 and COO minus, so now I'll have a negative charge. So I basically, going from left to right, I go from +1 to 0 to -1 as I change the pH. Okay? So you can imagine if this were in an electric field, at low pH, it's going to migrate one direction. At high pH, it's going to migrate the other direction. And this very basically helps illustrate how proteins function. If we change the pH, we change those electrostatic interactions, and that can have massive effects on binding materials or folding and stuff like that. Yes?
So you said pH is 0, the charge will be 0?
Yes, the charge will be
And
0 at the isoelectric point.
pK1 will be negative?
Yes, well, okay. So this is where it gets a little tricky. So let me go into this chart a little bit more. All the way down here, we basically have protonated everything, okay? So we're looking at just like one molecule, all right? Well, we got to think we got like millions or billions of these molecules in a solution. But if we protonated everything, it'll be +1. And from a theory, we look at one molecule, everything's protonated, my charge is +1. What's really happening at the pKa's is we're kind of 50-50. At this point, if we're at a pH of 2.34, that basically means we're half in the acidic form, so our COOH, and we're half in the conjugate base form, COO minus. So this is where we go back to that Henderson-Hasselbalch, my pKa, pH equals pKa plus the log of A minus over HA. The COOH is my HA, and the COO minus would be my A minus. All right. At the pKa, we're 50-50. Half of those sites are deprotonated. So we'd really say, right, this pKa, our charge is like +0.5. Okay, but that's getting too weird. We'll save that for like, advanced organic or something. All right. Similarly, our pKa2, this is where half of our amino groups would be deprotonated. So at a pH of 9.6, half of these would be NH3 and half of them would be NH2. We've removed about half of the protons at that point. If we keep adding base, we'll abstract more and more off, and eventually we get over here where they're all removed. So we get to that.
And the charge will be positive?
Yeah, sorry, the charge will be negative here because we're taking away protons. So the charge is positive at the highest pH because we've added all the H pluses we can, and negative at the other end. And then again, the key, one of the focuses on this particular graph is that PI. How do I know that the PI is 5.97? All right, so our isoelectric point. And this is really kind of neat. For glycine, it's not too bad, but we can make it worse. Yeah, which we will next week. So amino acids will have a charge of 0 at a very specific pH, and that's our isoelectric point. So, if I'm a little bit above that pH, then that means I'm going to have pulled some more protons away, so my solution, my molecules will overall have a slightly negative charge. If I'm a little bit below that pH, some more of them are going to be protonated than deprotonated, and I'll have a slightly positive charge. But right at the isoelectric point, my total charge is 0 on all my molecules in there. Okay, so how do we figure that out? Well, for glycine, it's just pK1 plus pK2 divided by 2. So if I go back up, I look at my pK for my carboxylic acid, 2.34, add that to 9.60 and divide by 2, and I'll get 5.97. Okay, all right. So at a pH of 5.97, the charge will be 0, no charge. It's free. Okay. This is when it'll also be the least soluble in water because there's no charge there, and it won't migrate in electric field. So that's actually kind of a neat way that you can do some protein separations in electric fields by changing the pH. So this is our simplest version. For glycine, we only have those 2 pKa's. And if we go one way, we become positive. If we go the other way, we become negative. Okay, not too bad. We'll make this more complex later on. What happens if you have like a side chain that's ionizable? Ooh, okay. All right. The other thing to keep in mind, and I sort of mentioned this, is that our amino acids can act like buffers. If you do DNA separations, like southern or northern blots, you'll probably make your gel using with a glycine buffer. Okay. So, glycine, we can have a pH of 2.34 or pH of 9.6. Plus or minus 1, basically we can make a buffer 1 pH unit above or below our pKa's. Okay. So our glycine can act as a nice little buffer in these two ranges, anywhere from 1.34 to 3.34, 8.6 to 10.6. Okay. We definitely cannot make a buffer with glycine around pH 6 because at our isoelectric point, you can see a very small change in pH is going to give you a huge change in overall charge. All right, so glycine, we know its side chain is just hydrogen. That hydrogen by itself is not ionizable. It's attached to the alpha carbon. But some side chains, like aspartate or asparagine, have ionizable groups as well. All right. So that basically gives us a third position, a third pKa to deal with. And we'll call that, for the ones that have those, pKr. Okay. So pK1 is always the carbonyl group. pK2 is always the amino group. And pKr will be the side chain if the side chain is ionizable. I believe in the text there's a nice little chart of the pKa values for every amino acid. I usually just kind of type it into Google real fast and pull up a chart. They don't always agree with each other, but they're always really close. So our ionizable side chains will influence the isoelectric point as well too, because now we've got three things to balance. So for like glycine, we're just juggling two balls. For asparagine, we throw a third one in there. We got to keep that balanced. Okay, so we can titrate those. We'll see that maybe in lab. The titration now gets a little bit more complex. Okay, so I think here we're looking at histidine, which is probably the easiest one to do. When we get to the amino acid titration lab, we'll talk about trying to titrate aspartate or something, which is a little more challenging. Okay, so if we look at histidine, our pK1 for our carbonyl group, all the way up here on the left. Okay, so below this, we're going to protonate that, and we'll have an overall charge of +2 because we have no charge on our carbonyl. We've got a +1 for our amino group attached to the alpha carbon, and we've got a +1 for the amino group on the side chain. So this is +2 here, right? And then this first proton that comes off is going to be the carbonyl group. So the carbonyl group attached to the alpha carbon is always the first proton that comes off, right? That's pKa is 1.82, comes off really easily, right? And we can see this would be our little buffer range here. We change our, keep going, becoming more basic, and we get to our pKr. Our pKrs are always in the middle. The side chain is always the next one to come off, no matter what it is. But this, now we're at in this region we're at +1 because we have negative charge and two positive charges. At pKr 6, half of this has come off. So now we start moving over here. We start taking off this one. So we get to this next form over here where our amino group, our alpha amino group is NH3+, COO-, and our side chain is deprotonated in neutral. So here our charge is 0. Okay. If I keep going, eventually I'll pull off this final proton here on that amino group. Its pKa is 9.7, and I can get a charge of -1. All right. So I've got 3 pKa's. Theoretically, I could make 3 buffers, 3 different buffer solutions from this. Okay. Now, here's the tough question, and this is what students often struggle with a little bit. So I'm trying to come up with better ways to get this across. How do we figure out the PI of this? And the temptation is, well, I've got 3 pKa's. I'll just average all 3 of them together, right? But that doesn't work. Doesn't work. What we really are averaging are the 2 pKa's where we go from 0 to +1 and 0 to -1. So in this case, my very first one, my overall charge is +2 and it goes to +1, still positive. So it's I'm still positive. So I'm not gonna count this one at all. Here, in the second case, when I go from my COO- to my side chain being deionized, I go from +1 to 0. So I want to use this pKa, which happens to be pKr. Okay? And then the very last ionization of the amino group up here, I go from 0 to -1. So I want to average these two together. If I add these two together, I get something like 16.17. Take half of that. My isoelectric point for histidine would be at a pH of 7.59 or 7.6, yeah, something like that. Something like that. All right. I guess you could say 5.8, 5.85, 5. Anyway, so we only average the two pKa values from 0 to +1 and 0 to -1. Okay. Does that make sense? Yeah. And we'll get more practice of this. Let me peek ahead of my slides here. Okay. Yeah. All right. Awesome. Okay. So in this slide just kind of illustrates exactly what I was saying there. We want to identify our particular species where we have a 0 charge. Okay. So in the case of histidine, it's where we've taken away 2 protons from the fully protonated form. Because if it's fully protonated form, I've got a +2 charge. I take away that first proton, I go to +1. I take away the second one, I get to 0. And I take away the last one, and I get to -1. Right? So I want to use these last 2 pK values, pKr for the side chain, and pK2 for the amino group. So, yeah. I guess we probably should have had this picture before the graph there. Okay. Questions about that? Okay. I always hesitate, so on Tuesday when we come back, I'll revisit this and I'll do a little board work and draw this out. Okay, because this is for an individual amino acid, right? But our proteins, we have hundreds, sometimes thousands of amino acids that are hooked together. So you can find an isoelectric point for, say, myosin or hemoglobin. What pH will it have no charge? And you just have to, if you have like thousands and thousands of ionizable groups, you find the one where it goes from +1 to 0 and 0 to -1 and average those 2 pKa's. All right. So let's just step briefly into that and talk about how our amino acids will polymerize to form peptides, because we're actually going to make finding our PI of a particular protein a little bit easier with this polymerization step. So basically what happens is the carbonyl group of one protein interacts with the amino group of a second amino acid, I should say amino acid in here. So we call this R1 and R2 for amino acid 1 and amino acid 2. But these two groups, carbonyl group of 1 interacts with the amino group of 2, and we have a dehydration reaction in here, or condensation. Right? So we basically, the hydroxyl group from the carbonyl group and the hydrogen from the amino group form water, and we get this nice little peptide bond in here. Okay. All right. Not too bad. Okay. When we do this, you'll note, hey, this is one of our ionizable protons from the carbonyl group, so we just got rid of it. And this is an ionizable proton from our amino group, and we just got rid of it. So we're kind of subtracting out 2 ionizable groups there. Okay. It's also worth noting, too, that our peptide ends are not the same. They're different. They have a directionality. We have an amino terminus, which will usually be positive, and a carboxylic acid terminus, which will usually be negative. Okay. So in this case, we have amino acid 1. We have to make a peptide bond. So here we have, was it serine? Ooh, yeah, look at the memorization here. And then glycine, and then tyrosine, and then alanine, and is that valine? I think that's valine. Might be isoleucine. Might be leucine. All right. So, yeah, it's leucine. Let me look at my notes. Okay. So, how would we name this compound? Ooh, that's terrible, right? Okay. There are a couple different ways we could name this. I could call this ceryl glycyl tyrosyl alanyl leucine, all right, which basically throws it up there. But that's a mouthful, and that's a pain, and that's hard to keep track of. Okay. I could use 3-letter abbreviations and call it, SER-GLY-TYR-ALA-LEU. And that's still a lot of letters, but that'll tell me exactly what each one is. But this is also why our proteins have 1-letter abbreviations as well too. Okay. So I would call this serine, S. G, tyrosine's 1-letter abbreviation is Y. A and L. So I can call it Seagal. I could pretend it knows Aikido and call it Steven Seagal. Ooh, good joke. Okay, TikTok that one. Okay. So this naming, again, go cerylglycyl tyrosyl alanyl leucine. This makes German look easy because there's always a joke about Germans have one word to describe something, but it'll be like 400 letters long. I remember I was studying in Austria, staying with the host family, and they had like a little girl who was in like 2nd or 3rd grade. So she liked to come over and do her homework with me when I was doing my homework for the classes I was taking. And at one point, she offered to help me, and I was like, oh, that's so cute, because I was struggling trying to read a newspaper article about traffic congestion because the traffic congestion word was about that long. So then she looked at it and she looked at me, and I could see her thinking, how do I explain to him so he'll understand it? And I was like, that was very humbling. So that's why I'm a biochemist and not a linguist. All right, but anyway, that's long, that's a mouthful, that's a pain to see. You can't instantly look at that and know what it is. This is a little bit better. Our 3-letter abbreviations explicitly tell us what's in here, but our 1-letter abbreviations are even better. SGYAL. Okay. All right. Do I want to do anything else with that? Where did I want to go here next? All right. Yeah. I think this is probably a good stop and place. We'll end about 5 minutes early today. But I think there's a link there to again, more flashcards for like Quizlet thing or something like that. Both these 3-letter and 1-letter abbreviations. I do want to say about why is tyrosine abbreviated Y? Well, T would be great, right? Except threonine is going to be T. So tyrosine, TY. Why? We'll use a 2nd letter in there, right? So leucine is L. So lysine. Anybody know what the 1-letter abbreviation for lysine is? It's K. Do you know why it's K? All right. Well, lysine can't be L because L is leucine. All right, well, what about Y, the 2nd letter? It's like, oh, nope. Y is tyrosine, so that one's taken. What about what comes after L? M. Nope, M is methionine. So, what comes before L? K. That's literally the reason for why lysine is abbreviated K, because that letter's not being used. There are lots of them that start with, L. See the same thing with like glutamate, glutamine, and glycine. Glycine is a G. Glutamate ends up being E. All right, and then glutamine is Q. Why? Because glutamine kind of sounds like Q-tamine. Craziness. All right. Okay, but that's it. Luckily, we have more letters than we have amino acids that you need to memorize. Okay. We'll pick up talking about peptides, start joining these amino acids together and making small chains, which we'll define as peptides. Sometimes you'll hear them called oligos. We'll talk about those functions, and we'll also figure out the PI of a very short peptide chain. Like, I think in the homework I have you do like a 4-peptide chain. If I do it on the exam, it might be like 2 or 3, so it won't be too awful for you. All right. Let's stop the screen sharing. Yes, yeah, you should probably, well, if I were better, I would make it a form fillable PDF for the lab, but I haven't done that. I haven't quite figured out how to put in all the fields yet for that. So you may want to print it out, but if you can actually have like a tablet and write on your screen or something like that, or a touch screen, you won't need to necessarily print it out. You can actually show me stuff like that. Because the way I'm going to have you guys submit it is to either scan it and put it in a Dropbox. Yes. Yep. And I'll fix that here momentarily.